potassium binds. Now you want to open it to the inside. You don't use ATP anymore. How do you change the shape back open to the inside? Just remove the phosphate. You remove the phosphate, the protein flips back to its original shape. So you only use ATP once. You use ATP at the beginning when you add the phosphate group. You don't need to use ATP again. Yes, ma'am. There is a hydrolysis reaction. After the potassium is released. To release the potassium, you have to flip the shape. So that's when the phosphate is hydrolyzed. This way. Where does what work? The phosphate is an inorganic phosphate, and it can be used by other reactions in the cell. So it's recycled. Very good question. This process is ongoing continuously in almost all animal cells, and especially in the neurons. Very important for the neurons to maintain the separation of the potassium and sodium at certain concentrations. And then when the sodium and potassium channels in the neurons open, you get flow of sodium and potassium down the gradients. So if you've ever heard of action potentials in neurons, the little spikes that you see, I will show you, they always show you on the electrocardiographs, for example. The little spikes come from action potentials that happen in both neurons and muscle cells. So we're not going to talk about how action potentials work in this class at all, but you have a little bit about that in your textbook if you're interested, and if you take by 1 or 2, they will spend more time talking about how neurons work. We wouldn't call it an activator. Good analogy, but not exactly. Because for the activator, we're talking about enzymes, then you have an activator that causes the enzyme to become active and carry out a reaction. So we wouldn't use that term in the phosphate gene. So when the protein is phosphorylated, that means the phosphate group is attached to it, it will cause it to change shape. And when it's dephosphorylated, it will change shape again. Activators do the same thing to enzymes. This is not an enzyme, so we don't exactly use the same terminology. But very good analogy. All right. So, of course, there are mutations in the sodium-potassium complex could be linked to some human disease as well. So, again, migraines. Again, if you remember that your neurons are dependent on these different concentrations of potassium and sodium in the cells across the membrane. And so, if the pump is not working properly, that could lead to problems, neurological problems. Migraines, one example. Paralysis on one side of the body called hemiplegia of childhood, for example, is linked to this. Dystonia of Parkinsonism, which is lack of muscle control, stiff movement, difficulties in swallowing and speech. I hope none of you ever come into contact with this potential problem because there's no treatment at this point. Now, one of the consequences of having these types of pumps is that your cells always have a membrane potential across the membrane. So that means that you have higher positive charges on one side compared to the other side. So typically, your cells are more negative inside than they are compared to the outside. So when you talk about voltage, it's always a difference across the membrane. One side of the membrane compared to the other side of the membrane. And your cells are always referred to as negative inside. So you measure the voltage inside compared to the outside. So in animal cells, you're talking about negative 60 or 70 millivolts at the resting phase. Plant cells, Gesundheit, about negative 110 millivolts. Bread mold, which is a fungus, negative 180 millivolts. So depending on different species, you can have different charges maintained across the membrane. So typically, you're looking at negative 60 millivolts difference across your cell membranes. So the distribution of the positive and negative ions across the membrane is what creates this voltage difference. And you can think that there are all kinds of ions that your body depends on: calcium, magnesium, potassium, chloride. So all of those have negative and positive charges, and some of them have higher concentrations on the inside, some of them have higher concentrations on the outside. So it's the balance among all of those ions that leads to what your overall membrane potential is. Now, When you're looking at the movement of ions across a membrane, whether you're looking at passive movement or active movement, you're always looking at the electrochemical gradient. If you're looking at something that is not charged, like a sugar molecule, you're only looking at concentration gradient, because that's all that matters. But if you're looking at an ion, the concentration matters, but so does the charge. Because if your cell is more negatively charged inside, what would happen if a negatively charged ion tries to come in? The negative charges repel each other. So if your cell is more negatively charged inside, negative 60 millivolts, chloride with a negative charge is going to have a hard time going in based on the charge difference. But sodium with a positive charge would have an easier time going in based on the charge difference. But charge isn't everything. You also have to consider the concentration gradient. Because there's usually more sodium outside than inside. There's usually more potassium inside than outside. So more potassium inside, diffusion would favor it to go out based on concentration, but the charge would favor it to stay where it is because it's positively charged. So it becomes more complicated when you're looking at ions as opposed to something that is not charged. Sugar molecules, concentration is enough for you to figure out if it's going to diffuse this way or that way. With an ion, you have to know both the chemical force, which is the concentration gradient, and the electrical force based on the charge differences and the electrochemical potential, the membrane potential of the cell. So we're going to do a couple of examples. But before we do that, I just want to remind you that pumps, like the sodium-potassium pump, are referred to as electrogenic pumps because they create a difference of charged units across the membrane. So if you go back and take a look at the sodium-potassium pump, how many sodium ions is it going to take from the inside of the cell? And it's going to move them to the outside. How many potassium ions is it going to take from the outside? 2. And it's going to bring them in. So it takes 3 positive charges out of the cell and brings only 2 back in. So in effect, what is it doing to the membrane potential of the cell? Yes, sir?
It creates a higher negative potential on the outside of the cell.
Higher negative inside or outside? If you're moving more positive charges outside and only bringing 2 inside, which side of the membrane is more negative? The inside is more negative. So the sodium-potassium pump makes the inside of your cell more negatively charged because it's removing more positive charges than it brings in. So we refer to these types of pumps as electrogenic pumps because they create a voltage gradient across your membrane. And that's going to be really important for the movement of ions back into the cell or further out of the cell, or if you're looking at how other pumps or other transport molecules might work. Now, in plants, fungi, and bacteria, you don't have the sodium-potassium pump. They have a proton pump, so they pump protons out. In animals, you have the sodium-potassium pump. 3 sodium go out, 2 potassium comes in. Plants just pump protons outside. And that's how they maintain a large negative charge inside of the cell. So let's take a look at an example together. I'm sorry, was that a question for that?
Yeah. How does the cell discriminate against any positive ion?
What do you mean discriminate?
Like, is any positive ion able to enter the cell?
So you have to go back to what we did at last week with the structure of the membrane. What can cross the lipid bilayer on its own? What can cross the lipid bilayer on its own? Yes, sir. Which are examples of what? In general terms, what can cross this lipid bilayer on its own? Small non-polar molecules like oxygen and carbon dioxide, which is the ones he mentioned, can cross the membrane on their own. Why is that? What is it about this membrane that prevents other things from moving in but allows O2 and CO2, which are small non-polar molecules, to move in? The fatty acid tails are hydrophobic, so nothing charged can cross this. So the membrane does not allow anything charged to go through. How do charged stuff get in? How do charged stuff get in? Facilitated diffusion, either through channels or through carrier proteins. All right, back to our example here. Plant cell, membrane potential, negative 100 millivolts inside the cell. You know the concentration of calcium is higher outside than inside. What will happen to the calcium? Take a minute, consider your options, then we'll take a look.